So good morning, students.
Good morning.
Very few students have joined. Half of the class has joined at this moment. Let's wait for just maybe 1 or 2 minutes. Let others join. So students, any question related to monoclonal antibody? We have started this topic yesterday. Okay, students, so before we start, let's have some questions. So, define monoclonal antibody, or what do you understand by monoclonal antibody? So, students, just raise your hands. What do you mean by monoclonal antibodies? Because today we will be talking about how monoclonal antibodies are produced. Okay, so but before that, you should know what is monoclonal antibody. Okay, so I can see one hand being raised, but I want more hands to be raised. So if it is not clear to you what is monoclonal antibody, then there is no use of having today's class, right? What will you do by producing monoclonal antibody if you don't know what is monoclonal antibody? So students, who will define monoclonal antibody? And yesterday everyone said it's clear. So you really want to understand, or should I just complete the topic and move ahead? So Manaswini, what is monoclonal antibody?
Sir, it's an antibody that's derived from a single clone and it's specific to a single epitope of a particular antigen.
Right, right. So yes, uh, it's derived from a single clone of cell, okay, and it is specific against a specific epitope of an antigen, right? So you have to specify this. It's because one antigen may have different epitopes. Okay, clear? Okay, I really don't know. Should I ask more questions because I did not get the answer for the very basic thing. Okay, can you name the two scientists who devised the method for uh, producing monoclonal antibodies? Anyone? Name the scientist who devised the method, and they also received the Nobel Prize for this work. Yes, Sanjana? Sanjana, you are not audible. No, Sanjana, you are not audible. Yes, Shakti.
Am I audible?
Shakti, what's the answer?
Am I audible?
Yes,
Yes. So the scientists were Kohler and Milstein.
yes, George Kohler and Caesar Milstein. Okay, so these two scientists they devised a method by which you can uh, get monoclonal antibodies, or you can prepare monoclonal antibodies in the lab. Okay, now my next question is to produce a hybridoma cell. Okay, uh, what two different types of cells are required? So just raise your hand. To produce a monoclonal antibody, we need two different types of cells. What are those two types? So Tanvi has raised her hand. Yes, Tanvi.
So B cell and myeloma cell.
Okay, so a B cell and the myeloma cell, right? Okay, uh, so which cell is providing what property to the hybridoma cell? So what is the need to fuse two different cells? Anyone? Yes, Tanvi.
So myeloma cells have the immortal growth property, and B cells have the genetic material for the antibodies.
Yes, so B cell provides the genetic information, right? Which antibody has to be produced against which antigen. So the information comes from the normal cell, okay? And the myeloma cell, it is cancerous and it secretes antibody, okay? So, and it also provides the immortal growth property because it's a cancerous cell, okay? So, when we fuse these two cells, right? So both the properties then come in a single cell. Okay. So now that single cell or the hybrid cell or the hybridoma, it have the information which antibody has to be produced. Okay. And it is cancerous and it is a plasma cell. So it keeps secreting the required or the desired antibody. Okay. Uh, okay. Okay. My next question is, what do we use to fuse the cells? What do we use to fuse the two different cells? Yes, Manaswani?
So we use polyethylene glycol.
Yes, PEG, polyethylene glycol. Okay. Okay. So now let me share the slides and again we'll start with the production of monoclonal antibodies. So what I really want today is that it should be clear to you, right? Because when you will see the application part of these monoclonal antibodies, you will realize how important these monoclonal antibodies are, right? So it's a very simple procedure, okay? Only thing is you just need to understand few things, uh, and the whole procedure will be very simple, okay? It may look complicated, but it is not complicated. It's very simple, okay? So let me just share the slides. Okay, so we have already seen this that George Kohler and Caesar Milstein, they come up with a method for preparing monoclonal antibodies, and the basic idea was to fuse a normal B cell, right, which provides the genetic information for antibodies, for the production of antibodies, and a myeloma cell. And we have already read about myeloma cell. It's a cancerous plasma cell. So the method was to fuse the two cells, make a hybrid, okay, and let that hybrid cell produce monoclonal antibodies, okay. But it was not as simple as it looks, because uh, when we fuse these cells, so many different combinations will come up, okay, and you have to just select the hybridomas, right? So this was the main thing, how we just let the hybridomas grow and not the other cells. And for this work, they also received the Nobel Prize, okay. And this again we have seen that we require polyethylene glycol, okay, for fusing the two cells, okay. So we use polyethylene glycol. This we have again discussed that myeloma cell will provide immortal growth property, and the B cell will provide the genetic information for antibody production. And when we fuse these two cells by polyethylene glycol, we get the hybrid cell, okay, or the hybridoma, okay. Now when we actually do this, the problem is that you get unfused myeloma cells, you will get unfused B cells, you will get combination of fused cells like the B cell, they can get fused, myeloma cells can get fused, and obviously there will be one combination of B cell and the myeloma cell which we actually need, okay. Now students, uh, B cells, right? So they are normal B cell, and you know every cell has a lifespan. Okay. So the B cell after its lifespan it will die. So we are not worried about the B cells. They will die. B cell B cell fused fusion, they will also die, because these are the normal B cell. They have a like lifespan, right? After which they will die. Okay. Now the problem is the myeloma cells, right? We don't want these unfused myeloma cells. So we need that they should also be killed, or they should die, right? So what we do is we take these myeloma cells and do a genetic modification in them, okay. We remove some part of the gene which produces for a particular enzyme, okay. So when that enzyme is not there, right, so these myeloma cells will die, right. So okay. So now there was a need to have a specific media which only allows growth of the hybridoma cells. Okay, and for this, a hat medium was designed. Okay, and now hat stands for hypoxanthine, aminopterin, and thymidine. Okay, and you will understand why hypoxanthine, why thymidine, and why aminopterin was added in this media. Okay, so in this media, okay, uh, B cells will not survive, okay, and they will die out. And because of the genetic modification, these myeloma cells will also not survive, but the hybridomas will survive. And this is what we need to understand, because the B cells contribute the missing enzyme for the salvage pathway. Okay, So as we discussed yesterday, there are two pathways. One is the de novo pathway, right, which the cell is using normally. Okay, so under normal conditions, a cell will use the de novo pathway, right? So under normal conditions, the cell will use de novo pathway for synthesizing nucleotides, right? So you can understand the importance of these two pathways. So when these nucleotides are formed, these nucleotides forms the DNA, right? I'm sure you have read the structure of DNA. You know what is nucleoside, what is nucleotide, how the DNA structure looks like, right? So if these nucleotides are not formed, the DNA will not form, right? And the cell will die, right? So hatch selection depends on the fact that mammalian cells can synthesize nucleotides by two different pathways. One is the de novo pathway, which is normally going on, okay? And when de novo pathway is not working, the other pathway, the salvage pathway, is used, okay? Now, in HAT, one of the component is aminopetrin, right? And what aminopetrin does? It blocks the de novo pathway. Okay. Now, de novo pathway is blocked by aminopetrin, right? So, in that condition, what will the cells do? Right? For the survival, they'll use the second pathway. The cells will now utilize the salvage pathway. Okay, clear till this point? So there are two pathways for nucleotide synthesis. One is the de novo pathway, one is the salvage pathway. And I don't know if in biochemistry, if you have read these two pathways, it is good. But even if you have not read, at least you should remember these two names. Okay. So now I've already told what aminopetrin does. It blocks the de novo pathway, right? So the cell will now utilize the salvage pathway. Okay. Now, which bypasses the aminopetrin block by converting purines and pyrimidines directly into nucleotides for DNA and RNA synthesis. Okay. So salvage pathway will uh, make the nucleotides by a different way. Okay. Now, de novo pathway is gone. So let's understand some important enzymes of the salvage pathway. Okay. So salvage pathway में there are two very important enzymes which play a role in this salvage pathway. One of them is hypoxanthine guanine phosphoribosyl transferase, which is also simply called HGPRT. Okay, so this is hypoxanthine guanine phosphoribosyl transferase. So this R is this. Okay, hypoxanthine guanine phosphoribosyl transferase, HGPRT. Okay, and the other enzyme is the thymidine kinase, TK. Okay, now these two are the most important enzymes of the salvage pathway, and if there is mutation in either of these two enzymes, right, so that will block the ability of the cell to use the salvage pathway, right? And if you remember what I said, that when we take the myeloma cell, we make some small modification, right? So we create mutation in these enzymes. Okay, so we create mutation in HGPRT and thymidine kinase. Okay, so students, I think now तुम्हें समझ आ रहा होगा de novo pathway is blocked by aminopetrin. Okay, तो दूसरा कौन सा पाथवे है? सैल्वेज पाथवे. सैल्वेज पाथवे के लिए कौन से एंजाइम्स चाहिए? HGPRT and thymidine kinase. What we are doing is we are creating mutation in these enzymes, right? So what happens? The cell will not be able to use the de novo pathway because of aminopterin, and it will also not be able to use the salvage pathway because there is mutation in HGPRT and TK genes, right? So the myeloma cell will die. Clear? B cell अपने आप मर गया, माइलोमा सेल की प्रॉब्लम थी, तो उसका सॉल्यूशन ये था कि एमिनोपेट्रिन डाल दो, इट विल नॉट बी एबल टू यूज द डी नोवो पाथवे, एंड उसकी जो जीन्स हैं उसमें म्यूटेशन कर दो of HGPRT and thymidine kinase genes, right? So it will not be able to use the salvage pathway also, right? So both the pathways are blocked, de novo is blocked, salvage is blocked, and the cell will die. Clear? Clear? Okay. So as I said HAT medium contains aminopterin to block de novo pathway. Okay. Hypoxanthine and thymidine are there to allow growth by salvage pathway, right? So what other things we have added in HAT medium is hypoxanthine and thymidine, right? And these two are required for the growth by salvage pathway. ओके, तो हमें सैल्वेज पाथवे चलाने के लिए भी तो कंपोनेंट्स देने पड़ेंगे मीडिया में, राइट? सो वी आर पुटिंग हाइपोक्सांथिन एंड थायमिडीन. ओके, सो देयरफॉर द सेल्स दैट लैक ईदर HGPRT और TK विल डाई इन HAT मीडियम. ओके, तो जिन सेल्स के पास HGPRT या थायमिडीन काइनेज नहीं है, they will die, right? Because they lack the ability to use salvage pathway. Okay, so ना de novo pathway चलेगा, ना salvage pathway चलेगा, and the cells will die. Okay, so B cell गया और myeloma cell गया. Okay, now still the question is, then why the hybridoma is surviving? B cell भी नहीं survive कर रहा, myeloma cell भी survive नहीं कर रहा, तो hybrid क्यों survive कर रहा, right? Okay, so let's understand this. Okay, this is just what I showed. There is de novo pathway. Okay, so this pathway, it is blocked by putting aminopterin. Okay, so this pathway is blocked in HAT medium. Okay, now the cell will use the salvage pathway. Okay, and as I said, the cell needs thymidine and hypoxanthine to use the salvage pathway, right? So if the cell have this thymidine kinase gene and it has the HGPRT gene, then it will utilize this pathway for the production of nucleotides and the cell will survive, right? But if we create mutation here in HGPRT or thymidine kinase, and if they are not there, right, so this pathway will also not work. Okay, clear? So students, clear?
Yes, sir. Yes, sir.
Yes, sir.
Okay, now coming to the most important part. So right, just see here, it's written TK minus, okay, and this is TK plus, right? So TK minus means that the gene for this is not present. Okay, or it is mutated, right? And so when we talk about uh, these myeloma cells, okay, what happens? We have already taken out the TK gene, right? So this dies, right? Because this cell is not able to use de novo pathway, this cell is not able to use the salvage pathway, right? Now, this B cell, the normal B cell, it has the thymidine kinase gene, okay? It has the thymidine kinase enzyme, right? But it has to die because it has a specific uh, time of survival, right? So after some time, the B cell will also die, right? But the hybrid of these two cells will survive because this B cell is providing the thymidine kinase gene to this fuse cell. So you can see here it has become TK plus, and from where this TK gene has come, it has come from the B cell. Okay, now this hybrid cell have the thymidine kinase enzyme. Okay, so this can now use the salvage pathway. Okay, and it will survive because it has the thymidine kinase enzyme and it can use the thymidine which we have already put in the hat medium, right? So we have hypoxanthine, aminopterin and thymidine. So we have put this component here and therefore this fused cell will survive. Okay, clear why hybrid cell is surviving and not the others? Okay, and you can either do mutation in thymidine kinase TK or you can do mutation in HGPRT. So you can either mutate HGPRT or you can use the TK mutant. Okay, I think this is very simple. Okay, students, so if you have any questions, you can ask. I'm taking attendance now, and after the attendance, I'll show you a video, right, to uh, show this process of monoclonal antibody production, right? So if you have any questions, you can ask. Is everything clear? Yes, Shakti?
Yes, sir. So, sir, if we are producing like hybridoma cells in large quantity, and if we have unfused myeloma cells in also large quantity, then we have to do a genetic modification of all of them in like separately.
Uh, yes, right? Because there are so many cells, right? And suppose if you just do it in one cell, only that will survive, right? Not all
Yes.
will survive. So if you just do it in one, two, or three, or five, or 10 cells, right? And just put them, others will die, only those mutants will survive, right?
Okay, sir.
Yeah. Any other question? And Shakti, it's not a big problem to mutate many cells, right? Because you are so there are a lot of ways by which we create mutations. Okay, so we can create mutation in large number of cells at one go also. Okay,
Okay, so it's not that tough.
yeah.
Thank you, sir.
Okay, so let me have attendance, and after the attendance, I will show you a video. Okay, so Adish,
Present, sir.
Atoiba,
Present, sir.
Ayushi,
Present, sir.
Sam,
Present, sir.
Devanshi, Ishita, Ishita,
Present, sir.
Janvi,
Present, sir.
Manaswani,
Present, sir.
Kshitij. So Kshitij is not there. Namrita,
Present, sir.
Nandini, Nandini, Nehal,
Present, sir.
Rupal,
Present,
okay, Sahiba,
present, sir.
Present, sir.
okay, Sahiba, you are not visible.
So I'm having connectivity issues today because of the rain.
Okay, Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridiv,
Present, sir.
Vivek, Vivek, Yukta, Yukta, Yuvraj,
Present, sir. Sir, I am present.
okay, Yuvraj,
Sir Vivek here, I am present.
okay, Vivek. Okay, so I think only Kshitij is absent today. Okay, students, let's see a video on hybridoma technology. Just see this, and if any question comes to your mind, then you can ask me, okay? So let me just share this.
About the hybridoma technology. So this hybridoma technology is used for the production of large number of monoclonal antibodies against a particular antigen. And these monoclonal antibodies are used for the different purposes. For example, in the treatment of cancer, and also as anti-venom, for example, anti-snake venom, anti-bee venom, anti-spider venom. Basically, these monoclonal antibodies are produced by a white blood cell, which is called as plasma cell. Whenever any antigen enters in the body, especially the extracellular antigen, the B cell or B lymphocyte get activated by it, and it is converted into the plasma cell. And finally, this plasma cell secretes the antibodies against that particular antigen. Now we might think, if the antigen is administered into the body of animal and that animal is going to produce plasma cell against that particular antigen which produces monoclonal antibodies against it, we can isolate this plasma cell from the lab animal and they can culture into the artificial media, and in artificial media this plasma cell will produce antibodies for us. It is correct. The plasma cells can be cultured into the artificial media, but the biggest problem is that the lifespan of plasma cells is very short. It is from few weeks to few months. And that's why we cannot produce antibodies for a long time from culturing them into the artificial media. In hybridoma technology, this problem of short lifespan of plasma cell is overcome by fusing it by a cancerous cell which is called as a myeloma cell. Myeloma cell, due to it is a cancerous cell, it is immortal in nature. So we have fused one cell which has the ability for production of antibodies and other cell which is immortal in nature. The new cell which is produced due to the fusion of these two cells is called as the hybridoma cell. And therefore this hybrid cell gains the property of both cells. So this cell is going to produce antibodies as well as it is immortal in nature. Let us see the actual procedure of hybridoma technology. First of all, the antigen against which the antibodies are to be produced is injected intravenously to the lab animal. This lab animal is generally mice or any other mammal. Since the antigen is given intravenously, that is directly into the blood, the antigens are trapped into the spleen. Spleen is the secondary lymphoid organ. Inside the spleen, the antigen interacts with the B cell and activates them to convert into the plasma cell. Once the plasma cells are produced, they will secrete the large number of monoclonal antibodies against the antigen injected. After this plasma cell producing the monoclonal antibodies against our injected antigen, they are isolated from the spleen. After the purification of plasma cell, it is fused with the myeloma cell. The fusion procedure is carried out in the presence of polyethylene glycol, and sometimes electroporation can also be used. Since the fusion procedure is random, it is not like that the plasma cells will always fuse with the myeloma cell, and therefore at the end of fusion procedure 5 different types of cells are produced, and they are fused plasma cells, fused myeloma cells, unfused plasma, unfused myeloma cell, and the most important hybridoma cell. Among these 5 cells, we are only interested in the hybridoma cell, and therefore the next step is the selection of hybridoma. Selection is carried out on the HAT media, HAT. Here the H is for hypoxanthine, A is for aminopterin, and T is for thymidine. These are the contents which are.
So students, इससे पहले जितना बताया everything is clear?
Yes, sir.
Yes.
Okay, good.
Are present in the HAT media. See, whenever any cell divides in the artificial media, before dividing, it must synthesize the new copy of the DNA. And for that, it requires the synthesis of nucleotides. Nucleotides can be synthesized by two ways. Either the cell can go for the salvage pathway or it can go for de novo synthesis. In case of salvage pathway, cells uses the degraded part of old nucleotide to produce the new nucleotides. And in case of de novo pathway, completely new nucleotides are synthesized using small metabolites like different sugars, different amino acids which are present in the media. In case of hat media, cells cannot operate de novo pathway, and this is due to the presence of aminopterin in the media. This aminopterin, it blocks one key enzyme which is required to operate the de novo pathway, and this enzyme is dihydrofolate reductase. Once this enzyme is blocked, cells cannot operate the de novo pathway. Okay. Therefore, there is only one option left for the cells in the hat media, and that is the salvage pathway. Cells can operate salvage pathway, but for this also there is one condition, and this condition is that cell must have one enzyme which is called as HGPRT, that is hypoxanthine guanine phosphoribosyl transferase. And for operating the salvage pathway, which have this particular enzyme, they can use the hypoxanthine and thymidine as precursor. Plasma cells have this enzyme, but myeloma cells don't.
Class.
I want to remind you that 5 different cells are produced after the fusion procedure of plasma and memory cells. And these are fused plasma, fused myeloma, hybridoma, unfused plasma, and unfused myeloma cell. Among these cells, plasma cells have HGPRT, and so the fused plasma cells. Myeloma cells don't have HGPRT enzyme, and therefore fused myeloma will also lack it. What about hybridoma? In case of hybridoma, one cell is plasma, and therefore it will have HGPRT enzyme. Correct? Now, what will happen if all these 5 different kind of cells are transferred on the hat media? Cells which lack HGPRT enzyme will not grow on the hat media, and these cells are fused myeloma and unfused myeloma. The other 3 types of cells can grow on the hat media, but lifespan of plasma and the fused plasma is very short, and within few weeks they will automatically die. No need to separately kill them. What about hybridoma again? It is HGPRT positive and it is immortal in nature. It will keep growing on the hat media. It has no limit. And therefore at the end hybridoma cells will survive on the hat media. In this way the hybridoma is selected and used for the continuous antibody production.
So students, I hope after this video, this technique of hybridoma is clear to you.
Yes, it's clear.
Okay, so any questions? Okay, so I think uh, it's clear to you now. So we have today discussed monoclonal antibodies. Okay, we have seen how they can be uh, grown in hat medium, how hat medium works, okay, what is the role of each component in this hat medium. Okay, so I think we have discussed everything related to this. And once you have grown your specific cells, okay, you can use them in cell culture and propagate it as much as you want, and you can harvest these, and then from that media you can take out the monoclonal antibodies, you can purify them, and then use it for various purposes. Okay, so as I said, one of the most important purpose is in diagnostics, right? So, whenever you go for a diagnostic test for any infectious disease, right? So, if they are using some serological methods, then they are most probably using a monoclonal antibody. Okay, so just for example, even yesterday I discussed that in ELISA we are using monoclonal antibodies, right? Uh, have you read the technique of Western blotting? Students, have you read Western blotting?
So we learned it last semester, but it was very brief.
Okay, so any idea what we do in Western blotting? Any idea? Okay, why do we carry out Western blotting? No?
So we carried out to detect like a particular protein from
Yes. Yes. So as you said, we want to detect a particular protein, right? So when you are targeting a particular protein, you need to have a specific antibody, right? Which binds only that particular protein. Okay, so suppose you take any sample. Suppose you have taken bacteria, right? And you want to find a specific protein in that bacteria. So obviously, when you will do the protein separation, right? So there will be a lot of different proteins. Okay, so how will you find your protein, the protein of your interest in those hundreds of different proteins? So what you will do is you will use a particular specific monoclonal antibody, and that monoclonal antibody will go and bind to only the protein against which it is specific. So you can get to know whether that protein is present or not in those hundreds of proteins. Okay, so this way it helps in detection of particular protein or drug, right? It can help in detection of infectious diseases, right? So suppose your blood is taken, okay, so the blood will be having either the antigen और द एंटीबॉडी, राइट? ब्लड के अंदर इन दोनों में से कोई एक चीज तो होगी, ठीक है? कई केसेस में क्या होता है कि पैथोजन नहीं होता है, so in that case you look for antibodies, right? And in some cases you can directly look for the antigen, right? So in both the cases, right, suppose you are looking for antigen, okay, so you require a specific antibody which will bind to that particular pathogen or that antigen, right? But even if you are looking for an antibody, you need to have a antibody which is against that antibody, right? So antibody against an antibody. So monoclonal antibody will be used even if you are looking for an antigen, even if you are looking for an antibody, if you are looking for a particular protein, right? Just for example, अभी COVID के time पे we just said that there is cytokine storm, right? So if in your body you want to see the concentration of cytokines, or you want to see the presence of particular cytokines, you can again use monoclonal antibodies, and through flow cytometry you can get to know the concentration of that particular uh, cytokine, right? Whether it has gone up, it has gone down. So flow cytometry is a very important uh, technique which we use in immunology, right? So I think I'll just stop here, and in the next class we will talk about the different applications of monoclonal antibodies, right? And there are many applications. It's being used for various purposes, okay? And we'll discuss that in the next class. So I hope this monoclonal antibody had technology is clear to you, and in the next class I will again uh, ask you to discuss this technique before we proceed for the applications of monoclonal antibodies. Okay, so with this I am ending the class now.
Thank you, sir.
Okay, students.
Thank you, sir.
Thank you.